Welcome to the video for Module 10, Part 2, Performance of Milk Assay using Plate Counts.
Okay, welcome back to Lab Module 10. We're going to be setting up a series of dilutions that will allow us to determine the number of bacteria in this sample of contaminated milk in this beaker. Let me just set the stage for you. So I have the contaminated milk sample here. I have a series of 10 tubes. You'll notice they have the red caps on them. These are going to be our dilution tubes. Each of these tubes contains 9 mils of sterile water. In addition, I have laid out 10 empty sterile Petri dishes. Also in front of me is a micropipetter set to dispense 1 mil of liquid, a box of sterile pipette tips that will be used with this micropipetter, of course a Bunsen burner, striker, and then a container into which I will dispose the contaminated pipette tips. Hopefully you watched the previous video where I spent a few minutes talking about pipettes and micropipetters and tried to explain the differences among them. If you haven't watched that video, I recommend you watch it first and then watch me use the micropipetter to carry out the dilutions for this lab module. Something else I have that's off screen because they need to sit in 55 degrees C water are 10 tubes of melted tryptic soy agar. These tubes of agar were melted and they are being kept at 55 degrees C so that they don't harden before I need to use them. There are 10 mils of liquid agar in each of those tubes. I'll just grab one of them out of the water, hot water, so you can see. You'll notice that the agar, melted agar tubes have the 10 mils of agar and have blue caps. So let's talk a little bit about what we're trying to do here in this lab module. As I mentioned just a couple minutes ago, the goal is to determine the number of bacteria contaminating this milk sample. Of course, I deliberately put bacteria in here so that we could do this exercise. I really don't know how many bacteria are in there. Oftentimes, when we want to count the number of bacteria in a sample like milk, we eventually want to come up with a number of bacteria, usually referred to as the number of colony-forming units, or CFUs, per milliliter of the original sample. In this case, the original sample is milk. So the question becomes, how do we do that? How do we take the contaminated sample and eventually determine the number of colony-forming units that are present in every milliliter of the original milk sample? Well, you might think, well, why not just spread them out on the surface of agar? You know, take a milliliter of milk, get yourself a tryptic soy agar plate that's sterile, but has the agar in it, of course, and then just dispense a mil of the liquid, a mil of the milk, onto the surface and then spread that over the surface of the agar. Well, you can do that. Technically, it's possible, of course, but it won't help you because there are probably so many bacteria in 1 mil of this milk that they would all grow together on the surface, and you would find that the surface of the agar would be completely covered by bacterial growth. That doesn't help us. We need to prepare our milk sample in such a way that when we do get it onto tryptic soy agar, the individual bacteria are so spread out that they form individual, isolated, easily counted colonies.
Here is a TSA plate with about 34 or 35 isolated colonies growing on its surface. Each of those colonies contains bacteria that are all descended from one or two original bacteria that were placed on the surface of the agar at that position.
Now, when you start with a sample of milk that is heavily contaminated, like this one is, there are probably at least hundreds of thousands, perhaps millions of bacteria per milliliter of milk. The only way you're going to get the number of bacteria to be small enough that you'll have a small enough number of colonies on a plate to count is by diluting the milk sample. And that's what this is really all about. I'm going to introduce you to what's called a serial dilution method that will allow you to accurately determine the number of bacteria in any liquid sample that you might run across. In this lab, the liquid sample, of course, is contaminated milk. So how does this work? Again, let me provide an overview of what I'm going to be doing here, and then I will take you through the protocol step by step. So overall, the idea is this. We take these 10 tubes of sterile water. There are 9 mils of sterile water in each of these dilution tubes. The volume is important because what we're going to do is we are going to use these tubes to carry out a series of 1/10 dilutions. We will make a 1/10 dilution into the first tube, and then we will make a 1/10 dilution of tube 1 into tube 2, mix, make a 1/10 dilution of tube 2 into tube 3, mix, make a 1/10 dilution of tube 3 into tube 4, mix, and so on until we have reached the last and 10th tube, which will represent a 1/10 dilution of the number 9 tube. That's the overall approach using these tubes of sterile distilled water. Once we have made the 10 dilutions, we then need to somehow get a sample of each of the dilutions into a petri dish containing agar. And the way we're going to do that is we're going to take a milliliter from each dilution tube and put it into its own tube of melted agar. Each tube of melted agar is sterile, contains 10 mils of tryptic soy agar. As I mentioned earlier, the agar is being kept at 55 degrees C, so it remains melted until we need to use it. You might say, why did I choose 55 degrees C? Why didn't I keep it at 70 or 80 degrees C? Well, the problem is you have to use the agar at a temperature that it will not kill the bacterium you're trying to count. And the bacterium in this milk cannot survive exposure to temperatures above 55 degrees C. So it's important that the temperature of the melted agar is no higher than 55 degrees C. Okay, let's go through the protocol. I'm going to put my sample of milk here on my left. Make sure all the plates are turned over. Now, I'm not going to do this in front of you, but what I have done already is I have labeled each of the dilution tubes. Remember, each of these dilution tubes contains 9 mils of sterile distilled water. I've also gone ahead and I've labeled 10 empty sterile petri dishes with the same dilution numbers so that there is a petri dish that corresponds to each of the 10 dilution tubes. Now I'm going to read through the numbers very quickly.
Please remember that the dilution numbers that I'm going to be reading to you during this video in the next few minutes are total dilution numbers. In other words, the first tube is a 10th dilution of the milk sample. The second tube is a 100th dilution of the milk sample. The third tube is a 1,000th dilution of the milk sample, and so on and so on.
Tube number 1 is 1/10. Tube number 2 is labeled 100th. Tube number 3 is labeled 1,000th. Tube number 4 is 1/10,000th. Tube number 5 is 100,000th. Tube number 6 is 1 millionth. Tube number 7 is 1/10 millionth. Tube number 8 is 100 millionth. Tube number 9 is 1 billionth. And tube number 10 is 1/10 billionth. So each of these tubes has been labeled with a different dilution number, going from 1/10 in tube number 1 to 1/10 billionth in tube number 10. And as I mentioned, I have labeled these 10 plates similarly. So there is a 1/10 plate for the 1/10th tube, a 100th plate labeled for the 100th tube, all the way down to the 10th plate. The 10th plate is labeled 1/10 billionth to match up with the dilution tube that is labeled 1/10 billionth. So let's go through the actual workings of the protocol. I am only going to demonstrate the first 2 or 3 dilutions on screen, and then off screen I'll finish the dilutions, and then I'll come back on screen to show you how we're going to transfer the samples from each dilution tube into tubes of melted agar, and then pour those tubes of melted agar into the correct sterile empty Petri dishes that are lying here on the bench. So the first thing I need to do is fire up my Bunsen burner. I take this micropipette and I verify that it in fact is set to pick up and dispense 1 milliliter of liquid, and it is. So for the first dilution, I need to load up a fresh, sterile pipette tip. You will know from the previous video, the one I asked you to look at first, that this box of sterile tips contains pipette tips that are blue and are designed to be used with this micropipetter. The top back on, the tip's out of the way for the moment. And now what I need to do is I need to use this pipetter to pick up a milliliter of the original sample and then transfer that milliliter to the very first dilution tube that is labeled 1/10, tube number 1. Now, I want to make sure this milk sample is adequately mixed. I gave it a good mix before I turned on the camera. But just to be sure there hasn't been any real settling out, I'm going to get a little bit of a swirl. I don't want to slop it on the bench because it is contaminated milk. Okay, it's probably adequately mixed. I take the pipetter, I put the tip into below the surface of the milk, I pull the trigger, and it automatically withdraws 1 milliliter, exactly 1 milliliter of the contaminated milk. I'm now going to take the micropipetter over to tube number 1, which is labeled 1/10. I take the top off the tube, I flame the mouth, and now I'm going to dispense the contents of this pipette tip, the contents being 1 ml. When I'm done dispensing, I dispose of that contaminated pipette tip into this container. I'm going to put the pipetter down, grab the cap, flame the mouth, put it back on. And then what I'm going to do is I'm going to thoroughly mix the milk that I added to tube number 1 with the 9 mils of sterile distilled water. The milk in tube number 1 has been diluted 1/10. 1 mil of milk plus 9 mils of sterile distilled water. I no longer need the beaker of milk. I'm going to move it out of the way so I don't accidentally tip it over. Next, I need to make a 1/10 dilution of tube number 1. I grab my box of tips, I take my pipetter, top off the tips, load the tip, the sterile tip onto the pipetter, top back on. Now what I'm going to do is I'm going to take tube number 1, which is the 1/10 dilution of the milk sample itself. I'm going to remove the top, flame the mouth, put the tip of the pipette tip below the surface, pull the trigger, and withdraw automatically 1 mil of the 1/10 dilution in tube 1. I'm now going to flame the mouth of tube number 1, put the top back on, and I'm going to put it near the back of the rack because I don't want to accidentally pick it up later on in the protocol. I now take tube number 2, which is labeled 100th dilution, remove the top, flame the mouth, and now I dispense the 1 mil that is in the tip into this tube. Dispose of the tip. I put the micropipetter down, flame the mouth of tube number 2, the cap back on, and now I'm going to mix tube number 2. Recall what I just did. I took tube number 1, which was a 1/10 dilution of the milk sample, mixed it. I transferred 1 mil from tube number 1 into the 9 mils of tube number 2. Well, 1 mil plus 9 mils is a 1/10 dilution. So what I did was I just now made a 1/10 dilution of tube number 1. But tube number 1 itself was a 1/10 dilution of the milk. So the total dilution of the milk in tube number 2 is a 1/10 times a 1/10, or a 100th, which is why tube number 2 is labeled 100. So at this point, I have prepared 2 dilutions. Tube number 1, which is a 1/10 dilution of the milk, and tube number 2, which is a 100th dilution of the milk. Let me do one more just so I'm sure you get the drift. So now I want to prepare dilution tube number 3 is labeled 1,000th. The way I'm going to prepare a 1,000th dilution is by taking 1 mil from the 100th dilution that I just did and putting it into the 9 mils in dilution tube number 3. So how do I do that? First thing I do is I grab my micropipetter, get the box of tips, load up a tip, close the box, make sure the pipetter is still set at 1. Of course it is. I now grab the number 2 dilution, the 100th, remove the cap, flame the mouth, put the pipette tip below the surface of the water, pull the trigger, automatically withdraw 1 mil of the 100th dilution, flame the mouth of tube number 2, replace the cap, put it at the back row. Now I grab tube number 3, which contains 9 mils of sterile water. I flame the mouth of the tube. I take the pipette tip, which has 1 mil of the 100th dilution. I am going to transfer that 1 mil into tube number 3. When I'm done, dispose of the tip, flame the mouth, put the cap back on, and now I have just prepared the 3rd dilution, tube number 3, which is labeled a 1,000th dilution. I make sure that I mix this thoroughly. Now I'm going to go ahead and do the other dilutions off camera, and then we'll come back and I'll show you how to handle the plates. But before I go off camera, let me just recap what we've done here to make sure you understand exactly how this is working. We started with a contaminated milk sample. We're attempting to determine the number of bacteria per mil, or actually, as your lab manual points out, the number of colony-forming units per mil of milk. We are doing this by performing what's called a serial dilution. To make the dilutions, we are using 10 different tubes, each containing 9 mils of sterile distilled water. What I did was I took 1 mil of the milk and I put it into tube number 1. I mixed, and at that point, tube number 1 represented a 1/10 dilution of the milk. I put the milk aside. I then transferred 1 mil of the 1/10 dilution into the 9 mils of the number 2 dilution. Mixed, put it down. To make the dilution in dilution tube number 3, I took dilution tube number 2, made sure it was mixed, and then I aseptically transferred 1 mil from dilution tube 2 into the 9 mils of dilution tube 3, and of course I mixed. Tube number 1 was 1 mil of milk into 9 mils of water. That's a 1/10 dilution. Tube number 2 was 1 mil of tube number 1 into 9 mils of water. That's a 1/10 dilution of a 1/10 dilution, which means tube number 2 is a total dilution of 100th. I then took 1 mil of tube number 2 and added it to 9 mils of tube number 3, which meant that tube number 3 is a 1/10 dilution of tube number 2. But we already calculated that tube number 2 is a 1/100th dilution, so 1/10 of 1/100th is a total of a 1,000th dilution. Hence the reason I labeled tube number 3 ahead of time as a 1,000th dilution. So I'm just going to continue now to finish off these dilutions. I'm going to go from tube 3 to tube 4, 4 to 5, 5 to 6, 6 to 7, 7 to 8, 8 to 9, and 9 to 10. Each of those transfers being a 1/10 dilution of the previous tube. So when I go from 3 to 4, I will take 1 mil of 3, add it to 9 mils in dilution tube number 4. So dilution tube number 4 will end up being a 1/10 dilution of tube number 3, but we've already calculated that tube number 3 is a 1,000th dilution. So the total dilution found in tube number 4 will be 1/10,000th. And I'll continue throughout the rest of the dilution series off camera. Okay, I'm back. I finished doing all 10 dilutions. The 10 dilutions are now in front of me. This is tube dilution number 1, 1/10. This is tube dilution 10, labeled a 10 billionth dilution. So how am I going to transfer the bacteria found in each dilution tube into agar that is going to be in 10 separate plates? Well, what I'm going to do in just a moment is I'm going to once again use my micropipetter and the pipette tips to transfer 1 mil from a dilution tube into 10 mils of sterile melted tryptic soy agar. Remember, the melted tryptic soy agar has been sitting here for a little while at 55 degrees C. That temperature will not kill the bacterium we're working with as long as the bacterium is not exposed to that temperature for more than a few minutes. Now, the volume of agar I'm using could be different. I could do 15 mils of agar or 20 mils of agar. I've had students in the past get a little bit confused about this. Since we use 10 mils of agar, they think that, oh, that's another dilution. It's not. The dilutions are all done. Whatever I do after this does not dilute the bacteria anymore or any less. All we're doing is we're transferring 1 mil from each of the dilution tubes into each of 10 different tubes of already melted sterile tryptic soy agar that's at 55 degrees C. It could be 20 mils. In fact, when I do this on my own, working in the lab, doing projects, I typically use between 15 and 20 mils of melted agar. I like having a little bit more. That way the agar doesn't dry out as quickly in the incubator. But we have found that 10 mils is usually enough. And so rather than waste materials, we have chosen to use the minimum that we need to use, which in this case happens to be 10 mils of melted tryptic soy agar. Okay, now I'm going to have to move quickly here because the agar will cool off quickly and then it will harden on us. And I have to make sure that I have enough time to add 1 mil of the dilution to 10 mils of agar, mix, and then pour the melted agar bacteria mixture into the first plate here and move it around to cover the bottom of the plate. I have to be able to do all that before the agar hardens. As soon as I take the melted agar out of the 55-degree water bath, it begins to cool, and I probably have less than a minute to complete that step. So here we go. I'm going to grab my micropipetter, get a new tip, top back on. Now what I'm going to do, and I'm going to move quickly here, is I'm going to grab the first tube of melted agar. As a blue cap, I'm going to put it here in the rack, near the front. I'm going to grab dilution number 1, the 10th dilution. I'm going to flame the mouth of that tube. I've already mixed all of these before I turn the camera back on. I'm going to pull up 1 mil of dilution tube number 1. Flame the mouth, the cap back on. Now I'm done with that one. Now to this still melted agar, I'm going to remove the cap, flame the mouth, and I'm going to add the 1 mil from dilution tube number 1. Dispose of the tip. I got to move quickly here. Flame the mouth, cap on, give it a little bit of a mix. All right, it's still melted. And now while it's still melted, I'm going to take the top off the plate and I'm going to pour all 10 mils of the melted agar into the plate. I'm going to take my plate and I'm going to move it around a little bit because I'm trying to get the entire surface covered. And it's a little tricky. Gotta do it before it hardens. Come on. Okay, looks like it has all covered the bottom. Okay, so what I just did, I took 1 mil from dilution tube number 1, which is the 10 solution. I placed it into 10 mils of melted tryptic soy agar. I mixed the agar, and I then poured the entire contents of that agar tube into plate number 1, which is labeled number 1, 1/10 dilution. I moved the plate around to make sure that the agar, while it was still liquid, covered the entire bottom surface of the plate. And now I'm going to put it over here and let it harden. Let's repeat that for dilution tube number 2. Dilution tube number 2 is a 1/100th dilution. I'm ready to go. I'm going to grab a tube of melted agar, put it in the rack, grab a micropipetter. I need to move quickly. Get a pipette tip. Grab dilution tube number 2. Make sure it's mixed. Pop the top. Flame the mouth. Take the fresh tip I have here. Automatically withdraw, pull the trigger, and automatically withdraw 1 mil of dilution tube number 2. Flame the mouth, top back on, put it at the back, and grab the melted agar tube, move the top, flame the mouth, transfer the 1 mil of dilution tube number 2 into this melted agar. Dispose of the tip. Mix this really well. And while it's still liquid, take plate number 2, which is labeled 1/100th dilution, and pour the entire contents of agar tube 2 into plate number 2. Tube back, top back on right away, and now swirl the contents of the plate to completely cover the bottom surface of the plate. And put it aside to harden. And that's it. I've done that for 2 tubes, dilution tube number 1 and dilution tube number 2. Off camera, I will process the remaining 8 dilution tubes.
1 mil of each dilution tube.
Will be placed into an individual tube of melted agar. The melted agar will be poured into the correctly numbered plate. So when I'm all done, I will have 10 plates labeled 1/10 dilution at the front end, at the left end, all the way to 1/10 billionth dilution at the far right end. Let them all harden, and I'll stick them in a 37-degree incubator to give them a chance to grow. I'll come back tomorrow. I will try to identify the plates that have between 30 and 300 colonies. Whichever plate or plates have a number of colonies that falls between 30 and 300, I will count the number of colonies. And then I will be able to use that number of colonies to calculate the number of colony forming units in the original milk sample. Now, something I did here, I did this for the purpose of the video, is I did only one plate for each of the dilutions. What I'm probably going to do off camera is I'm going to get another 10 empty plates and 10 more tubes of melted agar, and I'm going to go back to these 10 dilutions and make another complete set of plates. So that way I will have duplicates of each dilution on plates. I'll have 2 plates of the 2 plates of the 1/10th dilution, 2 plates of the 100th dilution, 2 plates of the 1000th dilution, and so on. You never want to do bacterial counts or plate counts on a single set of plates. You always want to have at least duplicates of each dilution and preferably triplicates, because you do get some bouncing around when you do replicates, and you need to have at least 2 plates to get an average from. So again, I didn't set it up that way for you to see. You'll just have to trust me that when I get off camera, turn everything off, I'm going to finish these plates, and then I will immediately set up another set of 10 plates to match the dilutions that we made. Well, that's it. Thank you for watching this video. Goodbye.